So the cancer is actually interacting with its environment to create its success. And again, that's not all tumors, but quite a few. This is a little cartoon of kind of what I'm driving at with that slide. This is, kind of a generic cartoon here. It'd be like something from the lining of your gut would be a good place where it starts out, right? So this would be the interior, the lumen of your gut, right? This would be the cells that will face that lumen, right? And some of this is the supporting tissue that's underneath it, right? You can notice from this that this little yellow band that's sitting down here, that's actually a series of proteins that are secreted by this tissue down here. This is called extracellular matrix, right? It sits down here. It's something that these cells out here can attach to. They will adhere. They will use that as a reference point of orientation for them. So what are some of these genes that I talked about? Well, if we're talking about this, over here in this kind of brown shaded region would be the cells that are undergoing uncontrolled growth. Right? They've sustained some kind of genetic damage, right, that allows them to outgrow their neighbors over here. And I should tell you as an aside, that what are the cells in this tissue? Normally, cells in the lining of your gut, they're being sloughed off. You lose them, okay? And they're constantly being replaced. So it is the cells that are at this layer that's closest to this little yellow band, right, that are the ones that are most actively dividing. As these cells are pushed out, right, towards the surface here, right, they lose the ability to divide. Normal growth control. Okay? So there is a series of cells down here that are normally dividing, but as they push their brethren out towards the surface, they lose that capability. These cells don't lose that capability. They've lost growth control. Okay? So that when these guys down here are dividing, well, you know what? They push out towards the surface and they keep on dividing. They don't stop. Okay? So this, can cause problems. This kind of tumor, it can overtake some of the tissue that's surrounding it. It's probably not going to be terribly bad. Certainly it can be removed surgically quite nicely. The next stage in development of this kind of tumor is that we'll penetrate this layer down here. Okay, so now we've gone through something that's uncontrolled cell growth, and we've caused some other genetic changes in this cell. And those genetic changes allow it to penetrate through this layer. Okay? So that now it's secreting certain proteins that digest this layer down here and allows it to move into the surrounding tissue. We call that malignant. Right? Still in one place, but it's malignant. Right? But it has occurred because of genetic changes, right? Not directly related to loss of control growth. Right? They're related to penetrate through this extracellular matrix. Finally, right, in the lifespan of the tumor, it's not unusual, it becomes metastatic. It moves around the body. So that once it penetrates into this layer down here, it has the potential, right, for entering into the bloodstream and being carried by the circulatory system to some other tissue where it will invade that tissue and begin to grow again. Okay. More genetic changes. Right. There are loss of developmental control. This stage down here where it gets to be penetrating into the vascular system, right, is actually a loss of developmental control. You can actually look at the genes that get expressed in these cells, and you can see a switch gets flipped. Right? Molecular switch gets flipped that actually allows these cells to penetrate through here, and it gets flipped back the other way when they try to invade the tissue and try to grow again. Okay? So a lot of little changes that are going on in these cells. And again, it's really quite horrendously complex process. Right? But most people, historically, a lot of research is focused on the loss of growth control. That's one of the big deals here in trying to attack a tumor, is generally speaking, try to attack its growth control. There are people out there researching, how is it invading the tissue? Can we block the metastatic phenotype of this? Can we block malignant phenotype? And yeah, I mean, there's some truth to that, but most of it's focused on, most of the research is going to be focused on control of growth. Okay, and that's basically what I've worked on for quite some time. So I throw this figure up here. It's a little more complex than what it probably needs to be. So what we're talking about is division of a cell, right? Getting one cell and getting it to divide into another cell. So this is just a way people diagram what changes a normal cell goes through when it is undergoing cell division. Okay, and it's blocked off into different stages. So obviously we have something over here called G1, S, G2, and M. Most cells are going to hang out here in G1. Most of the cells in your body are in G1. Some people like to draw this little GO out here. You can argue about that philosophically if you like. The bottom line is they're not actively undergoing division. Okay. Some cells are refractory to undergoing cell division anymore. They've lost the ability to do so. Some cells still retain, even in the adult, the ability to undergo cell division. Okay. So for those cells that can still undergo cell division, right, what we have out here in G1 is an ability to sense the environment. Right? What is the environment? Well, the environment is if I'm touching my neighbor. Right? The environment is if I'm receiving a signal that's a hormone telling me to divide or not to divide. There are some hormones that suppress division. There's all sorts of things. Nutrition. Do I have enough nutrients? Literally, is the cell big enough? Generally speaking, cells have to reach a certain size, a particular size, before they actually will be able to undergo cell division. So there are all sorts of signals that the cell receives, right, in terms of trying to figure out whether or not to divide. It's a balance, right, between positive factors and negative factors. The positive factors, things that are going to try to divide the cell, push it through the cell division cycle. Negative factors, things that are going to stop it for whatever reason. These are the two primary types of genes that we talk about when we talk about growth control. They are the two primary types of genes that we talk about when we talk about cancer research. Okay. We talk about those positive factors, hormones, growth hormones. You cut yourself, right? Your platelets rush in, right, to create the clot. And they burst open. And one of the things that platelets have, lots of growth hormones. They have all sorts of growth hormones. Why? Well, they're causing the cells that are in your skin to migrate and divide to seal the wound.